Hello. Can you all hear me okay? Okay. So, good morning. Y'all awake? Come on. Okay. Morning. So, my name is Katie. I am currently a junior in chemical engineering at Virginia Commonwealth University in Richmond, Virginia. And I'm going to talk about emulsion stability in microgravity. So, who here has ever shaken up salad dressing before they eat it? Raise your hand. I know you have. Come on. Okay, congratulations. You have created emulsion before. So, you might be wondering, what is an emulsion? So, an emulsion is when you have two immiscible liquids that are mixed together, and you have a dispersed phase and a matrix phase. So, usually, emulsions are characterized by their droplet size, and as you can see on the presentation, is between 0.5 micron and 10 microns is a typical macroemulsion. And on the slide, you can see three figures from literature, and on the left, we have an example of gelatin, which is a common food additive, and so many things like salad dressing that we talked about, or ice cream would be examples of emulsions in food. And then on the middle here is some research in the motor lubricant industry that we are working on in the lab. And those are some microscope pictures, which is also an example of an emulsion. And then on the right, you have an example for pharmaceutical targeted drug delivery that uses emulsions as well. And you can imagine that the stability of an emulsion is very important because, say, you were to get a drug delivered to your body and it wasn't the same concentration that it was supposed to be, that would be very problematic. So, it's very important that we understand the stability of emulsions. So, on the left, if I can figure out how to use the laser pointer, or not. So on the left, you can see we have an emulsion that's mixed up, and under the influence of gravity and other body forces, you can see creaming will occur, which is when the density differences cause particles or droplets to settle out or to cream upward. Then another phenomena that can occur is flocculation, and that is when droplets will come together to try to reach a more thermodynamically stable state and try to get larger. Then on the bottom, you can see there is Ostwald ripening, which occurs through diffusion. So the droplets and particles don't have to touch for this to occur. And what happens is the droplets will get larger at the smaller droplets' expense. Then you can see on the right, we have a surfactant and a polymer. These are different ways we can use to stabilize emulsions. Polymer can cause a physical barrier between the two droplets so they can't merge, and that is a phenomena called coalescence. So usually you have flocculation, which then leads to coalescence, which is the droplets merging together. Surfactants, which is one area that will be studied in this presentation, is when you have a molecule with a hydrophilic and hydrophobic ends, and they have electric charges that repel each other, which also prevents the droplets from merging. So as we talked about on the last slide, you have the effects of body forces such as gravity, and when you have those effects of gravity, you tend to have, you can see that droplets will get, the terminal velocity of the droplets changes and it scales with the radius of the droplet. So we can use, the effects of gravity, as you can see back here. When droplets get larger, they tend to cream or settle. And so when you have Ostwald ripening and flocculation occurring, typically there will be they will be masked by the gravitational or density effects. So it would be very difficult to study like flocculation or Oswald ripening on their own. So we can use surfactants and polymers to mitigate flocculation or Oswald ripening, but you can't really turn off gravity. So that's why we're going to study things in space. So under microgravity environment, you can see that droplets are smaller, and then you can see they're either undergoing Oswald ripening or flocculation, and then eventually merging to one large phase separation. And then on Earth, you can see the effects of gravity are typical. Yeah, everything tends to be masked by gravity. So you might be wondering, how do these samples get analyzed or made? So for sample preparation, we have a cell, and we put water mixed with surfactant into the cell, and then we add MCT oil, and then it is sealed with either a silicone or quartz cap and secured with bolts. To emulsify this sample, there will be a magnetic piston that is moved up and down, as you can see in that middle figure, and then an emulsion will be created. And on the right, you can see the experimental setup, and that is what the physical experiment looks like. That's our container, and it has the capability to have 4 experiments. So we're able to test emulsions in a very short amount of time after preparing all of the sampling. And so on Earth, as you can imagine, in space it would be very difficult to prepare a sample because there's microgravity. So all the samples are prepared on Earth and then sent up on the International Space Station to be tested. And so we can perform these SAM samples. Here we have the elegant breadboard, or EBB for short, and then in space there is a flight module, or FM for short. So we can run the same samples on Earth as like a control, and so you can see the effects of gravity, but it's very difficult to isolate Oswald ripening or flocculation because as droplets get bigger, they'll be under the influence of body forces more, so it's very difficult to study these rates independently. So these experiments will be run on Earth and in space. And so this is a short video that exemplifies you can see the coalescence and flocculation of droplets. This is in space, so there is no, it's under microgravity. So you can really see the droplets starting to go towards the middle. It almost looks like it's like sucking inward, but the camera's not moving, it's just your microgravity, and those are flocculation of particles and droplets. So we've talked about ways that emulsions destabilize, and you might be wondering how these emulsions can be measured. So the rate at which the destabilization occurs can be measured with diffusing wave spectroscopy, or DWS for short. DWS is a light scattering technique. So you can see on the left, you have a light source, typically a laser, will impinge the sample. It'll scatter through a droplet multiple times, which is one of the key significant differences for droplets and DWS versus other light scattering methods. And so then the light will come out on the other side. So you can see the APD, or avalanche photodiode, is used to measure the intensity of light that gets transmitted through. And as you can see in the figure, there's fluctuations in the intensity of light. And so this intensity of light, the fluctuations is a direct result of the droplet motion. And the intensity can be correlated using the G2 function, which is shown on the screen. This is a representative graph of the G2 function. So in the beginning, you have light that is well correlated. Let's see, is this? There we go. So you can see right here, the light is well correlated, and then as the droplets move, you start to have things decorrelate. So to get useful information from this, we go to a G1 function, which allows us to extract physical information. So as we talked about in the previous slide, you have your G1 function, and you can take the mean square displacement, which is this delta R squared, and it goes right here. And from this, you can get physical information such as A, the average A, which is the droplet radius, average droplet radius. And then over here we have L star, which is, as talked about in this previous slide, this the length between scattering events. And so as that occurs, you can see very likely that L star is increasing. It likely means that the space between the droplets is decreasing. And as your radius is increasing, that is very likely a causation of like flocculation. So here's an example of some real data from the International Space Station. This is our intensity function, and this is our correlation function. So future goals with the International Space Station include generating more L star and particle radius graphs, and to compare, basically to get the Oswald ripening and flocculation functions independently of gravity, so we can study more emulsions in the future without having to do expensive experiments. So I wanted to say thank you to all of the people who have helped with this. And quick questions, I know the time is close.
All right, thank you. So in your experimental setup, I was wondering, what are you guys actually changing, and what do you want to learn, and kind of what's the hypothesis
So one of the biggest things we're trying to study is the effect of Oswald ripening and flocculation, because under the influence of gravity, it's very difficult to isolate those by themselves, because as the droplets get bigger and larger, it's typically masked by gravity, so it's occurring at the same time, but it's very difficult to isolate. Also, we're studying different surfactant concentrations. So surfactants are used to stabilize emulsions. Studying that surfactant concentration. Yes?
Separate between Oswald ripening and flocculation. I'm trying to, what do we not understand about it? What's not known? Because I thought those processes are fairly well understood.
So we do understand that these processes occur, but we don't fully understand the rate at which they're occurring. So understanding how those influence emulsions independently of gravity is really what we're trying to understand. Does that answer your question, or are you still confused? Because I can explain it more. Okay. So.
If I have understanding of these models, I can maybe predict what's going to happen, and gravity is not a difficult thing to put in my model or remove from my model, and I'll see what happens. All that driven by surface tension and flows. So what am I not getting?
So under the influence of gravity, you can see flocculation and you can see Oswald ripening, but the effects of, once the droplets merge and get bigger over here, you can't isolate the effect of, say, creaming. So this droplet is going to get bigger, and then it's going to start moving upwards at the same time as it's flocculating together. So it's going to be very difficult to see the rate at which it's flocculating independently of the gravity. And these, like flocculation and Oswald ripening, typically they occur at a rate that's a lot slower than gravity. While they happen at the same time, it's just difficult to isolate the gravity from that.
And what you're trying to say is when flocculation or sedimentation occurs, it will be difficult to isolate the droplets and study the Brownian motion, because when they get packed or jammed, do you think this is the reason why it gets difficult? I mean, we're trying to figure out what is the difficult part. Is it the...
bigger part, droplets.
Well, they will get the bigger problems no matter what, Ed.
No, the droplets are bigger, so it's not a Brownian motion regime here.
Well, if the droplets get bigger, will you get out of the Brownian regime? And by the way, what is Brownian motion? Just so that...
So Brownian motion's like the random walk of how a particle moves within a mixture. So,
big
Oswald ripening.
big particles move like that? Like if I pick a 100 micron droplet, will it move under Brownian motion?
So all droplets are gonna go through Brownian motion. So this is
So there is no size effect.
for Brownian motion is just the effect of like the random walk of droplets. But so while Brownian motion, and you can see right here, we have a simple diffusion equation. While you have Brownian motion, it is not, like that is not what we're trying to study in this project. We're trying to study the effect of how droplets will coalesce. And so the importance of understanding how surfactants affect this and just how they behave independently is like the overall goal of the project. Yeah, sorry.